Thank you as well for having me here. I just wanted to provide you with a window that is a little bit outside of HIV and antiviral PK, and it's looking into early drug discovery and what we have been doing since the start of the pandemic. And this is in particular about the COVID Moonshot and ASAP discovery initiatives and what we have been doing in the space of protease inhibitors for what we can probably now call again a neglected disease. So I come from the Centre of Medicines Discovery at the University of Oxford, and our aim is very much to catalyse the discovery and development of novel medicines, and we're particularly focusing on areas where pharmaceutical companies generally don't spend their time. So we have a focus on mental health, a focus on rare disease, and then also focus on infective disease and other neglected targets. So I'd take you back 5 years, because prior to that, the Centre of Medicines Discovery was actually not working on infectious disease, but in contrast only on human targets. But at the start of the pandemic, the team at the Diamond Light Source, which is a big electron accelerator just south of Oxford, was contacted by a Chinese group saying, "We've got a new virus here. We've solved the structure already. Do you want to go and run a fragment screen on it?" And just to kind of explain what a fragment screen is, because this is something that probably doesn't come across your tables very commonly, it is something that takes into account all the structures that you see over there. So these are the different viral proteins that play a role in replication. And what you can do is basically get a molecular view of those structures and of the binding sites and see where different molecules bind. The trouble is if you use existing drugs, they will usually not bind to the molecule unless they are to that specific target. So it's not a good technique to kind of screen through big libraries and find compounds that are developed for a different disease for a novel target. What you instead need to do is you need to break up those drugs into very small parts so that they can bind to the active binding sites to then try and develop new drugs. And that's indeed what they've done. So they've taken the main protease. They also solved all the other structures and ran fragment screens, but that's not what I'm going to talk about today. I'm going to talk about the main protease because that was deemed as a very tractable and easy target to work on, and used a fragment library, or indeed several fragment libraries, to soak those little bits of molecules, fragments, into the active binding site. And that's what you can see in the left bottom corner. So that's the M-Pro structure. And you can see this very tightly populated site. That's the active binding site. That's where the protease cleaves both itself, but also other host proteins. The trouble with this is each and every single of those fragments won't really have any activity. So you could throw that on cells and it won't tell you anything. You have to put them together and grow them so that you can make a drug out of this. And it's a approach that is taken more and more commonly, especially for host-directed targets. But what the team at Diamond was then faced with was basically this pile of Lego bricks that they have somehow put together into a drug, but they don't yet know what might show activity both on the enzyme level, but then also on cellular level. So they did take an unusual approach. They went out, uploaded all that data onto a website and said, we call all the medicinal chemists worldwide that were mostly sitting at home because we were in lockdown, go ahead and design ideas based on those fragments, and then we'll make the best designs and test them in enzyme assays. And it really had an unprecedented uptake. It was retweeted at a rapid scale. We had thousands of designs coming in. Indeed, so many designs that we didn't really know how to move from these designs to a drug. But what did happen is we did crowdsource our ideas from the start, and that means that there is no IP attached to the drug, and that is, or any compound that gets developed. And that's really an unusual thought, especially if you think about downstream access problems that are often linked to patents and how these patents are distributed out. So this was an open science initiative from the start. But because of having so many designs, we had to kind of downscale to what we can actually physically make, because each of these compounds takes weeks to make in the lab. We're very glad to work with Enamine, which is a big synthesis company in Ukraine, and they went ahead and made these drugs at scale, but we whittled down to about 2,500 that we actually synthesized using AI route predictions, synthesis predictions, and then also binding modeling. And then came up and assayed all of them in high throughput assays, but also used high throughput crystallography to see how these compounds actually bind back into the binding site. And what is quite unprecedented in the kind of medicinal chemistry space is that we managed to get to something that was very active with potency, biochemical potency of about mid-30 nanomolar, and cellular potency around 6 nanomolar in only 18 months. And this is for completely new chemical matter that is a non-peptidomimetic inhibitor, which again then is important for the PK downstream. This is just an example of how we did this kind of fragment growing. So this was the kind of, on the left, the initial fragment merge, and then we kind of elaborated based on that with a drop in potency both on biochemical and enzyme level, and then ultimately also cellular level. What was exciting about this data, it was published in Science if you want to read more about the story, is that it ended up in being funded for preclinical development together with Drugs for Neglected Disease initiative that you referred to earlier by the Wellcome Trust, that we collaborate very, very closely with, but also through the NIH as one of the big antiviral pandemic preparedness avid centers later on. And I spend a little bit of time on each of those stories because they both offer some learnings. So the compound was great until we put it into animals. So it had excellent antiviral efficacy, had like a superb specificity, no activity on the other proteases. We were super excited about it. And then this was kind of the first measurement in blue, and basically it just doesn't stick around in mice. It gets rapidly eliminated. And just to kind of work on that, we had to kind of take some approaches because we thought this was a rodent-specific issue, because all our human measurements in human cells showed us that actually this compound should perform very well in humans. So initially, because we knew that this was a CYP3A4 substrate, we used a CYP3A4 inhibitor as a co-dosing strategy to just get the levels up so that we could perform some in vivo efficacy. So those are the kind of curves that you see that push the exposure up a little bit. So what we were really aiming for, based on previous studies on Paxlovid and that were published, was an exposure over free exposure over 3 times EC90 for at least 12 hours so that we could dose twice a day in our animal experiments. So that was one approach. And the other approach we took was in collaboration with the Drug Discovery Unit in Dundee, who have developed a humanized mouse model, which is actually really nice because you have to dose much less compound. They replaced all the different CYP enzymes, rodent CYP enzymes, with human enzymes, as well as PXR and others. And that really provided a kind of boost in exposures, and we managed to get over 12 hours of exposure. These are the linked efficacy results in an animal study, where we infected with SARS-CoV-2, and then on the left-hand side co-dosed with ABT on each day. On the right-hand side, we just dosed twice daily, and you can see that the exposures for the ABT co-dosing were not quite long enough. So we definitely needed exposures over 3 times EC90 for a continuous duration. As soon as you drop under that 3 times EC90, even if it's just for 1 hour, you don't see an impact on lung viral loads anymore. However, for the humanized mouse model, we did see a drop in lung viral loads as measured on day 3 on the right-hand side. We then had enough confidence to move into exploratory toxicology studies, so we scaled up the compound and gave it at high doses, 100, 300, and 1,000 mg per kg in rodents, and that's where we then had the next surprise. And what you can see here is the area under the curve plotted across the different animals, and you can see we get really nice dose response on day 1. There's a bit of a gender disparity in there, but on day 3 we basically have no exposure at all. And that was a real surprise because our initial induction experiments that we had done didn't show us any touch of induction, but we only screened in humans. And this was a rodent-specific issue. We didn't see the same effect in dogs. In dogs, we had a very nice exposure all the way through. So we would expect that we see something similar in humans based on the cellular data. However, we kind of tried to find a preclinical species, so moved back into mice, and there we did see exactly the same effect, where basically our exposures were completely abolished on day 3. And this is very, very rapid. Usually you see these kinds of lack of exposure after 2 weeks of dosing, and not only 3 days. So we went back and wanted to kind of find out what was going on, because if we wanted to develop this further, we really needed to nail down on what the mechanism of action is. So we kind of thought about formulation problems, did some long-term formulation studies that all seemed to be fine. Again, kind of pointing back to the dog data actually being okay. And then we reran the nuclear hormone receptor activation trial, and then found PXR-linked induction across all species, and went further into kind of cryopreserved human hepatocytes, just to kind of see what we kind of have in terms of effect on actual CYP levels. And we looked at both mRNA activation for different CYP enzymes, and indeed we do see induction on mRNA level and also catalytic activity level of CYP3A4. So this was indeed a CYP3A4 linked issue through PXR. So at that point, we had taken the decision to discontinue this particular compound due to the induction issues. It was actually a series-wide issue, so all the other compounds that we tested within that series showed the same effect. We we were not actually concerned about the human dose, but about how we would run reprotox study and downstream toxicology in the preclinical evaluation. But in addition to that, this was a narrow spectrum inhibitor. We only had activity against SARS-CoV-2 and SARS. We actually had equal activity against SARS, so we could have developed this exact inhibitor 20 years ago. But went ahead, put the results in a preprint, and at that time we had already moved on. We did have some learnings on the open science aspect of these studies, though. One was that external researchers extensively used all the data that we put out there, and that was for training purposes. They downloaded all our data sets, because preclinical data sets in that depth are not very readily available. The second one was that the company Shionogi actually explicitly cited our pharmacophore as an inspiration for Incitravir, a drug that's now on the market, which we thought was a perfect readout for an open science initiative, actually inspiring a novel drug. And then the third one was that our preprints and actual publications were extensively published and cited. The other very positive outcome was that we were approached by an NIH program officer pointing us to the novel AVID centers, which were big investments in antiviral research, and basically asked us to apply. And we did, and we're one of the 9 different AVID centers that were funded. So these were large-scale investments made by NIAID. And this was originally conceived as a 5-year collaboration to develop novel antivirals for pandemic preparedness. And we basically took a lot of the international consortium and crowdsourced consortium and asked our partners whether they would be interested in applying with us, and added a couple of work packages on. One, which was very important to us, is to think about building in resistance to potential downstream mutations from the start and the design of the drugs. And that's the work package 1, and I'm going to talk a little bit more about that. But all the other kind of essential parts, so the target enablement, the kind of structural biology that we use to get to our compounds, were kept the same. And then, most importantly, thinking about access downstream, we vowed to keep an open science angle with releasing all our data open access. But due to some of the conversations that we had with funders, we decided to develop a novel IP policy that is designed to enable open and equitable access by just defensively patenting a very small number of compounds that still gives other companies, if they are interested, the space to develop around, like very much like Shionogi has done, but gives the entity that wants to develop the asset downstream still a defensive strategy, and that has been published in Wellcome Open Research. So we went ahead and worked on 3 different classes of viruses: coronaviruses, enteroviruses, and flaviviruses, and that was very much based on the considerations that these 3 classes all had proteases, 1 or 2 different proteases. In the case of enteroviruses, we actually worked on 2, but we also extended beyond and worked on other targets that are less explored. So, for example, the N protein, endonucleases, and polymerases, where we also had some early stage projects ongoing. This is coming back to the idea of wanting to kind of design fitness costs or mutation costs into our compounds right from the get-go. And this is something that you have the beauty of, or the kind of luxury of doing if you're working with structure-based data. So we partnered with Matt Bojo and Carla Kierkegaard at Stanford, and they ran large-scale deep mutational scanning analyses where they basically mutated every single amino acid in the protease active binding site to look at the fitness costs of each amino acid change. And then in addition to that, we also used circulating, so phylogenetic data, and mapped that back onto the structure. And the idea then is, when you look at the right, that you design your fragment mergers away from the mutational tolerant sites. So you basically go for sites where any mutation would result in abrogation of protein function. And that kind of goes back to a term that was coined by Celia Schiffer in the HIV field, which kind of implicates that if you stay within the substrate envelope, so very tightly within the binding site, you have a much lower chance of resistance mutation. So that was the idea behind it. I mean, ultimately, we won't know whether it worked until any of our drugs makes it to the clinic. But we kind of thought that there must be a way of designing in some resistance or kind of some way of thinking around viral resistance. This is an overview of our projects, our most advanced ones. As you've seen, we've got more, and I'm going to talk more about the kind of broad spectrum protease inhibitor for a little longer, but we also just published on our broad spectrum flavivirus inhibitor that has activity across dengue, Zika, and West Nile. And we are working very hard on some enterovirus inhibitors where we are aiming to have activity against poliovirus, enterovirus D68 and A71, as well as Coxsackie virus B3. So we used DNDi 6510, so a compound that we discontinued with the known problems around the rodent-specific induction and the lack of broad-spectrum activity to kind of come up with some ideas. And what we were particularly interested in is developing activity against MERS, because currently there are no compounds available that would target MERS at therapeutic doses. And there also haven't been any kind of trials run. However, it is a disease of pandemic concern because it has a very high lethality. And what is particularly concerning that there's a high range of, there are lots of individuals living in close proximity to the animal reservoir. So with SARS-CoV-2 replicating in the patient cohort and spillover cases, there's a high likelihood of recombination and then a new circulating coronavirus that is potentially more contagious or lethal. So this was one of our main objectives. And Ed Griffin, he's our lead medicinal chemist within the ASAP consortium, looked very closely at the structure and basically identified 2 routes to develop into kind of conserved areas, but also to get some MERS activity, which is moving into the P1 prime pocket and to the P3 pocket. I usually have about 15 slides describing his medicinal chemistry strategy, but I'll spare all that and summarize it in like one little compound. This is basically the difference what we came up with after having made almost 2,000 compounds in 2 years of work. We basically exchanged the P3 pocket with heterocycle and then grew into the P1 prime pocket. And that really gave us a real change. So from having no efficacy against MERS, we did drop down to nanomolar efficacy against MERS in cellular assays. But in addition to that, even though we didn't originally aim for that, we did test our compound against a whole range of different animal coronaviruses in a reporter-based assay, and were able to show that at least at a high concentration, we do have activity against a whole lot, and that's in comparison to nirmatrelvir, which is the active component of Paxlovid. And this is, again, just a snapshot of a MERS local infection animal experiment where you overexpress human DPP4, which is the entry receptor just in the lung. And again, you can see that we basically completely abrogate viral load replication in the lung at low doses. And that, again, is in comparison to nirmatrelvir. And we're currently repeating this experiment with a follow-on compound that addresses the PXR risk better in comparison to ibuzatrevir, which is the optimized nirmatrelvir follow-up compound used by Pfizer in clinical trials at the moment. I can't be on a PK conference without at least including one slide on a human dose prediction. We used both allometric scaling and PBPK. Actually, Andrew Owen's group at Liverpool helped us very kindly with that. And what we were at this very early stage, so this is very, very early preclinical development, but what we were excited to see just as a kind of guidepost is that with the current data that we have, we are at once daily dosing, and this is for an acute dosing phase of 5 days, as previously for coronaviruses, around 100 mg per kg if we want to exceed 3 times EC90, so that's a kind of conservative range. But for the less conservative range, if you consider pandemic preparedness, you want to kind of have a drug in the pocket where you would, if you have less efficacy against that new circulating virus in the future, you still have some therapeutic margin so that you can dose up. So that's why we used a kind of more generous frame with 10 times EC90. And there we are still within a kind of reasonable dose once daily. And actually, we now ran toxicology experiments on this compound, which gives us a huge therapeutic margin on these compounds. The other thing that we were very excited about, which is not very common with peptidomimetic protease inhibitors, is that because we are non-covalent, non-peptidomimetic, we achieved brain exposure, at least in rodent, at very low doses. So this might, again, be a kind of coronavirus compound that can address a viral reservoir in the brain. So in summary, we selected a preclinical candidate that we're going to declare through DNDi in 2 weeks' time. So there will be some press releases coming with broad-spectrum coronavirus activity and in vivo efficacy against MERS and SARS. We got a human dose prediction that we are very happy with for this early stage of development. And we have got an extremely clean in vitro off-target panel, Ames negative and in vivo micronucleus negative, which is important for downstream genotoxicology. And very, very recent exprotox studies showed excellent exposure and an abrogated induction risk. So what we had seen with the previous compound is now designed out of this compound. In addition to that, and I didn't go into detail on this, is we have implemented this fragment screening technology as something that can be used for a wide range of viruses. And we now have a flavivirus compound that shows very nice broad spectrum activity in lead optimization, and an enterovirus compound that we are actually very excited about because it specifically shows polio activity. Which is a new thing, and a polio therapeutic would be very much needed, especially in phases of war and vaccination, lack of vaccination. But what we also regard as a very important outcome, even though our funding was cut short due to the NIH funding cuts or discontinuation of the AVID centers, that we were able to release all our discovery data open access in FAIR repositories for everyone to use. And the IP policy, which you can see over there, is published as well, which we hope will give a downstream path that we can use to develop these drugs for global equitable access downstream, especially in the global south. And But as you can see, most of the discovery partners are actually in the global north. So thanks for kind of sparking that discussion. It's a very good thought. And a big thanks to all the open science partners that have contributed, and especially the funders, in particular Wellcome Trust and NIH. Thank you.